Biomedical importance of carbohydrates. Carbohydrates are widely distributed in plants and animals. They have important structural and metabolic roles in plant. Glucose is synthesized from carbon dioxide and water by photosynthesis and stored as starch or used to synthesize cellulose of the plant framework. Animal can synthesize carbohydrate from glycerol and amino acids, but most animal carbohydrate is derived from the plants. Glucose is most important carbohydrate. Most dietary carbohydrate is absorbed into the bloodstream as glucose and the other sugar are converted into glucose in liver. Disease associated with carbohydrate metabolism include diabetes mellitus, galactosemia, glycogen storage disease, and lactose intolerance. Study of sugar's function in health and illness is known as glycobiology. Glycome is an organism's total complement of sugar, whether they are free or in more complex compounds. Glycomics is the study of glycomes in its whole, including genetic, physiological, or pathological and other aspects. It is like genomic and proteomic, the constituent sugar sequence and the connection of oligosaccharide chain encode biological information. The classification of carbohydrates. Starting with monosaccharides. Monosaccharides are those sugar that cannot be hydrolyzed into simple carbohydrates. They may be classified as triose, tetrose, pentose, hexose, or heptose, depending on the number of carbon atoms, 3 to 7, and as aldoses or ketoses, depending on whether they have an aldehyde or ketone group. Polysaccharides are condensation products of 3 to 10 monosaccharides. Most are not digested by human enzyme. Disaccharides are condensation product of two monosaccharide unit, for example lactose, maltose, isomaltose, sucrose, and trihalose. Polysaccharides are condensation product of more than 10 monosaccharides unit. Example are starch and dextrin, which may be linear or branched polymer. Polysaccharides are sometimes classified as hexosanes and pentosanes depending on the constituent monosaccharide, hexoses or pentoses respectively. In addition to starch and dextrin which are hexosane, food contain a wide variety of other polysaccharides that are collectively known as non-starch polysaccharides that are they are not digested by human enzymes and are major component of dietary fiber. Structure of glucose. Here you can see three types of structure of glucose. Number one is the straight chain form. Number two is alpha D glucose, which is Hayward projection. And number three is alpha D glucose in the chair form. The straight chain structural formula can account for some of the properties of glucose. Cyclic structure is favored on thermodynamic ground and account for the remainder of its chemical properties. For most purposes, the structural formula is represented as a simple ring in perspective as proposed by Hayward. Six-membered ring containing one oxygen atom is in the form of a chair. Sugar isomerism. D and L isomerism. A sugar isomer's identification as D form or mirror image as L form is decided by its special connection to the carbohydrate's parent component, the 3-carbon sugar glyceryl aldehyde. Whether a sugar belongs to D or L series is determined by the orientation of H or OH group around the carbon atom close to the terminal alcohol carbons or carbon 5 in glucose. You can see some structures of L-glucose and D-glucose. Sugar is the D isomer when the OH group is on carbon on the right, and sugar is L isomer when it is on the left, which is highlighted in the blue. Majority of naturally occurring monosaccharides are D sugar and the enzymes that break them down are specialized to this structure. Optical isomers. The presence of asymmetric carbon atoms also gives the compound optical activity. When plain polarized light passes through an optical isomer solution, it rotates either to right, which is dextrorotatory, or to left, which is levorotatory. Because the stereochemistry of sugar has no bearing on the direction of rotation of polarized light, it can be labeled as D negative or positive, L negative or positive. The D-negative isomer of fructose, for example, is the naturally occurring form. Glucose is dextrorotatory in solution. Thus, glucose solutions are sometimes referred to as dextrose. Pyranose and furanose ring structure. Ring structure of monosaccharides are like the ring structure of either pyran, a six-member ring, or furan, a five-membered ring. For glucose in solution, more than 99% is in the pyranose form. Pyranose and furanose form of glucose are given on the screen, and as well as the pyranose and furanose forms of fructose. Alpha and beta anomers. Ring structure of an aldose is hemiacetal since it is formed by the reaction between an aldehyde and alcohol group. Similarly, the ring structure of ketose is hemiketal. Crystalline glucose is alpha D glucopyranose. Cyclic structure is retained in solution, but isomerism occur about position 1, the carbonyl or anomeric carbon atom, to give a mixture of alpha glucopyranose 38% and beta glucopyranose, which is 62%. Less than 0.3% is represented by alpha and beta anomers of glucofuranose. Epimers. Isomers differing as a result of variation in the configuration of OH and H on carbon atom 2, 3 and 4 of glucose are known as epimers. Biologically, the most important epimers of glucose are mannose, epimerized at carbon number 2, and galactose, epimerized at carbon number 4. You can here see the alpha D galactose and alpha D mannose and the OH group on number 4 in galactose and on number 2 in carbon. Aldoses and ketosis isomerism. Fructose and glucose have the same molecular formula, but fructose has a possible keto group in position 2 and the anomeric carbon, whereas glucose has a potential aldehyde group in position 1, anomeric carbon. Aldoses are reducing chemical that are frequently referred to as a reducing sugar. By reducing an alkaline copper solution, this offers the basis for a simple chemical test for glucose in urine in poorly managed diabetes mellitus. Monosaccharide physiological importance. Pentoses of physiological importance. Pentoses mean 5 carbon sugars which include ribose, ribulose, arabinose, xylose, and xylulose. Ribose sources nucleic acid and metabolic intermediate. They are structural component of nucleic acid and coenzymes including ATP, NAD, and flavin coenzyme. Ribulose is metabolic intermediate in the pentose phosphate pathway. Arabinose is from the source is from the plant gum, constituent of glycoprotein. Xylulose, also from the plant gum, proteoglycans or glycosaminoglycans, constituent of glycoprotein. Xylulose is the metabolic intermediate excreted in the urine in essential pentosurias. Hexoses of physiological importance. Hexoses means six carbon sugars, which include glucose, fructose, galactose, and mannose. Glucose sources is fruit juice, hydrolysis of starch, cane or beetroot sugar, maltose, and lactose. Biochemical importance include the main metabolic fuel for tissue, blood sugar, excreted in urine in poorly controlled diabetes mellitus as a result of hyperglycemia. D-fructose comes from fruit juice, honey, hydrolysis of cane or beet sugar, and inulin. enzymic isomerization of glucose syrups for food manufacture. They are readily metabolized either via glucose or directly. Hereditary fructose intolerance lead to fructose accumulation of hypoglycemia. D-galactose comes from hydrolysis of lactose, readily metabolized to glucose, synthesized in mammary glands for synthesis of lactose in milk, a constituent of glycolipids and glycoproteins. Hereditary galactosemia as a result of a failure to metabolize galactose lead to the catarrhate. D-mannose, hydrolysis of plant mannan gum, constituent of glycoproteins. Sugar form glycosides. Glycosides are generated when hydroxyl group of monosaccharides anomeric carbon condenses with a second substance which might be another monosaccharide or in the case of a glycon, not a sugar. O-glycosidic bond is an acetyl link because it comes from reaction between hemiacetal group and another OH group if second group is likewise a hydroxyl. Resultant chemical is glycoside if hemiacetal portion is glucose, a galactoside if hemiacetal portion is galactose, and so on. An N-glycosidic bond is formed between adenine and ribose in nucleotide like ATP if the second group is amine. Biomedical importance of some sugar. Deoxysugars lack an oxygen atom. Deoxysugars are those in which one hydroxyl group has been replaced by hydrogen. An example is deoxyribose in DNA. Deoxysugar L-fructose occurs in glycoproteins. Deoxyglucose is used experimentally as an inhibitor of glucose metabolism. Then we have amino sugar. Hexosamines are components of glycoprotein, gangliosides, and glycosaminoglycans. D-glucosamine, a component of hyaluronic acid, D-galactosamine, also known as chondrosamine, a component of chondroitin, and D-menosamine are among the amino sugars. Several medicines like erythromycin contain amino sugars which are essential for antibiotic activity. Maltose, sucrose, and lactose are important disaccharides. Disaccharides are sugar composed of two monosaccharide residue linked by a glycosidic bond. Physiological importance disaccharide are maltose, sucrose, and lactose. Hydrolysis of sucrose yields a mixture of glucose and fructose called invert sugar. Because fructose is strongly levorotatory and changes, which mean inverts the weaker dextrorotatory action of sucrose. Next is polysaccharide storage and structural functions. Starch is a homopolymer of glucose forming a alpha glycosidic chain called A-glucosane or glucan. It is a most abundant dietary carbohydrate in cereal, potatoes, legumes, and other vegetables. Two main constituents of amylose, 15 to 20%, which has a non-branching helical structure, and amylopectin, 80 to 85%, which consists of branched chain composed of 24 to 30 glucose residues united by 1 to 4 linkage in the chain and by 1 to 6 linkage at the branch point. Glycogen is the storage polysaccharide in animals. Glycogen is more highly branched structure than amylopectin with chain of 12 to 14 alpha-D-glucopyranose residue in alpha 1 to 4 glycosidic linkage with branching by means of alpha 1 to 6 glycosidic bonds. Inulin is a polysaccharide of fructose found in tubers and roots of dahlias or artichokes. It is readily soluble in water and is used to determine the glomerular filtration rate. Dextrins are intermediates in hydrolysis of starch. Chitin is a structural polysaccharide in exoskeleton of crustaceans and insects, and in mushrooms it consists of N-acetyl D-glucosamine units joined by beta 1,2,4 glycosidic linkage. Cellulose is a chief constituent of a framework of plant. It is insoluble and consists of beta-D-glucopyranose unit linked by beta-1,2,4 bonds to form long straight chains strengthened by cross-linked hydrogen bonds. Cellulose cannot be digested by mammals because of the absence of an enzyme that hydrolyzes the beta linkage. Carbohydrates found in glycoproteins include acetylhexosamine, pentoses, methylpentose, and salicylic acid. Glycoproteins or mucoproteins are protein containing branched or unbranched oligosaccharides chains, including fucose. They occur in cell membranes, and many proteins are glycosylated. The sialic acids are N or O acyl derivatives or neuraminic acid. Neuraminic acid is a 9-carbon sugar derived from menosamine, an apimer of glucosamine and pyruvate. Sialic acid are constituent of both glycoprotein and gangliosides. Carbohydrate, cell membrane, and lipoprotein. In addition to lipid of cell membrane, approximately 5% is carbohydrate in glycoprotein and glycolipids. Their presence on the outer surface of plasma membrane has been shown with use of plant lectin, protein agglutinins that bind with specific glycosyl residue. For example, concatenavalin A binds alpha glycosyl and alpha menosyl residue. Glycophorin is a major integral membrane glycoprotein of human erythrocytes and span the liquid membrane, having free polypeptide portion outside both the external and internal cytoplasmic surface. Carbohydrates chain are only attached to the amino terminal portion outside the external surfaces. Then we have the summary of the carbohydrates of physiological importance. In today's video, we saw the glycome is the entire complement of sugar of an organism, whether free or present in more complex molecule. Glycomics is the study of glycome, including genetic, physiological, pathological, and other aspects. Carbohydrates are major constituent of animal food and animal tissue. They are characterized by type and number of monosaccharide residue in their molecules. Glucose is most important carbohydrate in mammalian biochemistry because nearly all carbohydrate in food is converted to glucose for metabolism. Sugar have large number of stereoisomer because they contain several asymmetric carbon atoms. Physiological importance of monosaccharide include blood sugar and ribose, an important constituent of nucleotide and nucleic acid. Important disaccharides include maltose, an intermediate in digestion of starch, sucrose, important as dietary constituent containing fructose and lactose in milk. Starch and glycogen are storage polymers of glucose in plants and animals respectively. Starch is the major metabolic fuel in diet. Complex carbohydrates contain other sugar derivatives such as amino sugars, uronic acid, and sialic acid. They include proteoglycans, glycosaminoglycans, which are associated with structural elements of tissue, and glycoprotein, which are proteins containing oligosaccharide chains. They are found in many situations, including the cell membrane. Oligosaccharide chain encode biological information depending on their constituents of sugar and their sequence and linkages. This brings us to the end of physiological importance of carbohydrates. Hope you like my video. Don't forget to subscribe for more. Thank you for watching.